Hello.
Hello, Danish sir.
Hello. Hello.
હા ડેનિસ સર. હા મારે ખાલી પૂછવું હતું કે આજની યુટ્યુબની લિંક અલગ નહીં હોય ને?
અલગ છે.
અલગ હોય એટલે પેલા બધાને મેલ એવી રીતે જ કરવો પડશે?
ના મેડમ, આના પર અહીંયા જ મૂકવાની. શું કરવા મેલ કરવાનું આટલા બધા જણાને? અહીંયા મૂકી દેવાની આપણે ચેટમાં. અને મેડમ સતત વેઇટિંગ રૂમમાંથી બધાને એડમિટ કરવા પડશે.
મેડમ આપણે આટલા જલ્દી કેમ એડમિટ કરીએ છીએ બધાને? હજી તો 5 મિનિટ બાકી છે.
10:58 થઈ મેડમ.
હા એટલે 11 થઈ 11:05 કરીએ ને. આપણે લેટ તો નથી ને?
શરૂ કરવાનું હોય ને મેડમ. 11 એ શરૂ.
હા પણ વાંધો નહીં, હજી મમતા મેડમ આયા નથી તો આપણે એડમિટ કરવા માટે 5 મિનિટ રાહ જોઈએ ને.
Madam, everybody will stay muted. It's okay. We'll wait till Mamta madam comes. There is no problem.
Danish bhai?
Madam, you can ask in chat. In chat, privately, you go to IT help desk name and there you ask in chat whatever you want to ask. Okay, ma'am.
Good morning, Dr. Prema Kumar. Sir, you are visible, but your mic is mute.
Okay, I unmute the mic now.
Yes, we can hear
Now.
you, sir.
Where is Professor Mamta?
She is joining us very soon.
Okay, right. Great.
I'm Alpana Shukla from MG Science Institute, sir.
Nice to meet you, Alpna.
Madam, let me know when I should start and we'll be starting.
I think we'll wait 5 minutes, then we'll start.
Sure, ma'am.
Yeah. Okay.
We request all the participants to keep their videos and mics off.
हैं? क्या? नहीं नहीं, दही हो तो रहने देना, और क्या। दही ज्यादा हो तो कटोरे में निकालो, तपेली घिसने दे देना।
Good morning, Chabarya, sir.
Very good morning, ma'am.
Good morning, sir.
Good morning.
Good morning, Dr. Mahesh.
Good morning.
From Sri Lanka.
Good morning. Very good morning.
Priti ma'am, you may request once again for muting their audios and keeping their videos off.
All the participants are requested to please their audio mute and their video off. Please make sure it is off while you have joined here.
Hello, is Mamta joining us?
Yes, ma'am is here. Alpna ma'am is joining. Shall we start?
Yes, please start.
A very good morning to all the respected guests, speakers, organizers, dignitaries, and delegates. On behalf of the LM College of Pharmacy and MG Science Institute, Ahmedabad, I welcome you again to the second day of our 2 days international webinar on medicinal botanicals, present and future trends, jointly organized by LM College and Botany Department, MG Science Institute. Before we proceed towards the scientific session, I would like to inform the participants that the link for feedback form will be given today in the chat box before we end the last session, that is at 1 p.m. sharp. Please make sure that you write your names in the feedback form correctly as you want it on the certificate, because your certificates will be generated automatically from the feedback details and will be available to you as soon as you fill up the form. So feedback form link will be available for half an hour only, that is between 1 to 1:30 p.m. So make sure that you fill it up between that duration.
Excuse me, ma'am, 1 minute. Suppose 100 participants have filled up and suppose they get the message that today you cannot fill up, then they must fill up that same link tomorrow.
They should inform us. We'll open the link for them if they are having some kind of issues.
Yes,
we
ma'am.
can open it if there are some genuine issues. Sure, ma'am. Now let us start with today's scientific session. We have with us our first speaker, Dr. G.A.S. Prema Kumara, who is going to deliver a talk on phytochemistry and applications of Sri Lankan medicinal flora. It gives me immense pleasure to introduce Sir. Dr. Prema Kumara obtained his PhD in Natural Products Chemistry and Chemical Biology from University of Colombo in 1996 and also received his postdoctoral training in Nutrition Biochemistry and Health Food Biotechnology in Japan in 2001 and 2 under STA Postdoctoral Fellowship and JSPS Invitation Fellowship. He received his Charter in Biology in 2004 and Charter in Chemistry in 2012. He is a fellow of the Institute of Biology and the Institute of Chemistry of Sri Lanka and an elected fellow of the National Academy of Science of Sri Lanka. After his PhD, Sir joined the Natural Products Development Group of CISIR Sri Lanka.
Excuse me, Preeti madam, we cannot hear you.
Preeti, you are muted.
Am I audible now, ma'am?
Yes, yes, please.
Okay.
Could you please go back to the beginning? We would like to know more.
Okay, yeah, ma'am. So Dr. Prema Kumara obtained his PhD in Natural Products Chemistry and Chemical Biology from University of Colombo in 1996 and also received his postdoctoral training in Nutrition Biochemistry and Health Food Biotechnology in Japan under STA Postdoctoral Fellowship and JSPS Invitation Fellowship. He received his Charter in Biology and Charter in Chemistry in 2004 and 2012 respectively. Sir is a fellow of the Institute of Biology and the Institute of Chemistry of Sri Lanka and an elected fellow of the National Academy of Science of Sri Lanka. After his PhD, Sir joined the Natural Products Development Group of CISIR Sri Lanka, now called as Industrial Technology Institute, in 1997 and pursued his career as a scientist. He became the Additional Director General R&D in 2012 and then served as the Director General for 5 years. Currently, Sir holds the chair of Industrial Technology Institute appointed by HE, the President of Sri Lanka. In parallel, Sir holds the position of Head of Department of Basic Sciences and Social Science at the Faculty of Nursing of University of Colombo, Sri Lanka. He is also a visiting academic to Southern Cross University, Australia. Sir held several key positions in national and international as well as ministerial committees, governing bodies, and has contributed immensely to the academia and the R&D sector in Sri Lanka. He has been appointed by HE, the President of Sri Lanka, to the National Research Council and to the National Committee on Women in Sri Lanka. Dr. Prema Kumara is a recipient of various presidential awards, National Science and Technology Award, ITI Best Innovative Technology Award, Professor Chandrasena Memorial Award of Institute of Chemistry Sri Lanka, Talented Scientist Award from University of Tirupati, Vetro World Innovation Finalist Award 2016, CARP Sri Lanka National Award 2019, Senate Award for Research Excellence 2019, and several fellowships including prestigious JSPS Invitation Fellowship of Japan. He has over 350 national and international research publications and scientific contributions, 2 books, 4 book chapters, and 2 commercial patents. Sir has nearly 30 years of academic and research experience in the area of bioactive natural products. Under his R&D projects, he has produced 9 MScs, 2 MPhils, and 15 PhDs under his supervision. Sir is a leading multidisciplinary researcher in Sri Lanka. He is a member of several learned societies in the country. He is a fellow of Institute of Biology and was the president of Institute of Biology Sri Lanka in 2007 and 8 and also founder chairman of Ethics Review Committee of IOB. With such an impressive CV, we are highly obliged to have you here today, sir, and we extend a hearty welcome to you. May I now invite Dr. Prema Kumara, sir, to please start the session. Sir, over to you.
Thank you, Dr. Preeti. Thank you, Dr. Preeti, for your very kind introduction. Can you all hear me?
Yes, sir.
Yes, we can.
Yeah. So, yeah, Dr. Mahesh, Principal of LMCP, Dr. B.K. Jain, the Principal of MG Science, Professor Mamta, other invited speakers, learned audience, fellow academics, the scientists, and the students. Good morning to all of you. And I must thankful to the organizers for inviting me to this important event. And I hope you all had a very fruitful session yesterday. And I hope I will be contributing through my speech for the knowledge and the betterment of this field. Thank you very much. And good morning to all of you. Can I share my slides now?
Yes, sir. Yes, sir.
Okay, do you see my slides now?
Yes, sir. Yes, sir. It is
Yeah, so yeah, may I start now?
sure, sir.
Yeah, So yeah, my topic is phytochemistry and applications of Sri Lankan medicinal flora. As explained, I'm attached to the University of Colombo, Sri Lanka. Let me show you where I'm coming from. As you all know, the Sri Lanka is the immediate neighbor of India, and I am located in Colombo. And this is my university, University of Colombo. This is the Faculty of Science where I graduated in the late 80s rather. And this is the place where I do most of my research work. And this is my faculty of nursing. And it is the only faculty in the country for nursing. And I head this department of basic sciences and social sciences. And this is the CSIR of Sri Lanka. And I happened to be a scientist there for a quite long time, and then moved back to university in 2018. And this is the new addition to CSIR, the Modern Research and Development Center, which is located in suburbs of Colombo. Actually, this is completed and during my tenure as Director General of this institute. So that is the background to me, basically where I'm from and what I have been doing is, of course, the Preeti explained well. And let me go ahead with my presentation now. So as you all know, the chemistry of plants and microbes played a very pivotal role in shaping up of this world that we see today. And from a barren, basically a very barren land, to a very lush greenish terrain, what we have now is actually evolution, and the chemistry of plants and microbes played a very important role in this transition over this evolution. And as you all know, the phytochemistry has been involved in almost all processes of the nature from the beginning of life on this earth. And the documentation of these processes by the scientific community has revealed the enormous contribution of phytochemicals to the betterment of this world. Yeah, let me close this background. Okay, sorry about it. So if you look at the basis of herbal-based medicines, the medicinal plants and medicinal microbes provided the base for all traditional herbal-based medical systems in the world. And as you all know, the chemical sciences in the 19th century uncovered that the curative basis of the herbal-based medicines are the phytochemicals therein, which has curative properties and the anti-infectious properties. So the current science that we practice today demonstrated the phytochemistry in these medicinal organisms are able to influence the biochemical and metabolic processes, and also the genetic makeup of an individual to restore or maintain the health of that individual. So if you look at the traditional medicinal practices in Asia, it is existed from time immemorial, as we all know. The Ayurveda, of course, dates back to about 1600 to 4500 BC, whereas the medicines, the herbal medicines that practice is date back to the same period, because of course the Bangladesh was part of India earlier. And we in Sri Lanka, we have a system called Deshiya Chikitsa and Hela Vedakama, which is different to Ayurveda, but it has been there as traditional system of medicine in this country, and which of course dates back to more than 2400 BC. But there, as you all know, there's a claim that we had a king called Ravana, which dates back to about 8000 BC, and he himself is a doctor, and he brought up this Hela Vedakama in this country. But we have no proper documented information with respect to this. But the archaeological information provide that this Hela Vedakama anyway dates back to about 2400 BC. And you have then the traditional Chinese medicine, or TCM, of course, again dates back to about 300 BC. And Kampo in Japan, of course, it's after the BC, of course, it is 500 AD. Sowa Rigpa in Bhutan is 700 AD. Jamu in Indonesia is about 800 AD. And Thai medicine, which is relatively young, is about 1200 AD. So the traditional medical practices in Asia, basically, the systems have broadly similar historical experience. Now, if you take Chinese, Arabic, Japanese, and Indic systems of medicine, they have attracted more research attention than any other traditional systems of medicine in the world. So the Chinese medicine, or Sonqi, or the traditional Chinese medicine, as we all know as TCM, is of course now called as Chinese medicine and pharmacotherapy, because it has got a greater share in the global herbal and medicinal market at present. And also we see that China has incorporated their traditional medicine into institutionalization or into modern institutions in the past century. And so the research on Chinese medicine, or TCM, is happening at a phase in this century. And as we all know, the Indian subcontinent too are involved in R&D on medicinal plants and traditional medicinal formula, and is taking place mainly in India, and the R&D inputs from India is also substantial. And when it comes to Sri Lanka, we too have a considerable contribution to the natural product chemistry of medicinal plants, and also involved in validation of traditional formula for its efficacy and toxicity. So that is the background to the Asian traditional medical systems. For me to start with the phytochemistry part of my speech. So when it comes to the South Asian region of Asia, now the South Asian region has been known for its medicinal biodiversity, and also long-standing tradition of these medical uses when you compare to the other part of Asia and the world. So we are in a way very fortunate to have these traditional systems of medicine well established, with a very highly diversified flora and fauna with medicinal properties. If you take Sri Lanka, we have 3,770 species of higher plants, whereas 897 species are indigenous, which is about 28.3% of endemicity. And we have about 1,414 species out of these 377 higher plants as medicinal. So it is about 37.5%. Now, of the medicinal herbs, about 200 species are extensively used in preparation of Ayurvedic and traditional medicines. As you may aware, although we have these medicinal plants in cultivation as well as wild harvesting, but still we depend on India for most of our medicinal plant requirements. So we import about 60% of our requirement from India. And though we are one of the biodiversity hotspots in the world, with the highest ranking in the biodiversity per 10,000 square kilometers in Asia, we have not actually have started organized cultivations to the extent that we need to use in our medicinal needs. And also, as you see now, we have a very remarkable high proportion of endemicity. It is about 25 to 28%. If you look at India, of course, I don't need to tell you, since you all are expert in this area, but as a completion, I have included India also into my talk. And India has 17,000 species of higher plants, whereas 7,500 are known for medicinal uses. So which is a remarkable proportion. It is about 44%. And if you take China, they have quite a large number, 30,000 high plants, whereas 10,608 are medicinal, which is about 35%. If you take Malaysia, they have about 15,000 species of higher plants, whereas the number of medicinal plants so far known, I would say, is about 2,000 plants. But of course, out of these 15,000, there must be many more medicinal plants, although they have not uncovered as yet. And of course, to about 2,700 species are endemic to the Malaysian peninsula. So now the beauty of this medicinal system is that what we practice in Asia, they are, of course, due to our literacy and ability of having these literate societies over many centuries. So our region, the Southeast Asian region, gave an extra edge to our medicines to be incorporated into well-documented system of medicines. Further, now the text provides a strong theoretical foundation and explanation to position the clinical and pharmacological experiments in this region. If you take Sri Lanka, this knowledge on this ancient medicine and also on the food habits what we have in this country is a gift actually from our ancestors to the present the generations, because it carries a wealth of traditional knowledge. So those are documented, and some are, of course, in folklore. And we have quite a number of different texts, which this our traditional system of medicine is documented. There are about 10 different texts that carries this traditional knowledge, where we can read and understand very easily. But at the same time, we have a set of quite a number of Ola leaf-based knowledge, which is actually with the traditional doctor families in this country, those who own those traditional, the recipes which are written down in the Ola leaf text. So if you have been to this country, you must have seen the archaeological marvels that we find in this country, ancient, stupas and, ancient cities and Buddhist temples and so on, all over the country, which shows that we have a very advanced technologies related to our living day-to-day life, and also in the medical medicines and the food patterns. And we are a country with over 3,000 man-made seasonal tanks in this country. These are seasonal tanks. Those are man-made seasonal tanks. So we have over 3,000 man-made seasonal tanks, so that we conserve our water to a great extent, the rainwater, and we use for our day-to-day activities. And also to tell you, we record the world first hospital in the north central area of the country. So that is a hospital the world ever known. The well-organized hospital was there in this country. And also to tell you, some of our kings, they themselves are doctors. So there are books that are written by these king on the medicine that they practice in this country. And at the same time, the archaeological excavations revealed excavated some surgical tools that are actually, when we compare with the present day surgical tools, those are very similar to the surgical tools. These excavated surgical tools have similarities to the present day surgical tools, as you can see. So Sri Lanka has a separate ministry for indigenous medicine and a department of Ayurveda. So they are mandated. So they are mandated for the development of the indigenous system of medicine, including training, registration, research, hospital care, and production of Ayurvedic drugs. Now we have about 3 teaching hospitals, 124 central dispensaries, 47 provincial hospitals, 231 local dispensaries, 1 research institute, 1 National Institute of Traditional Medicine, and 5 herbal gardens under the purview of Ministry of Indigenous Medicine. So to tell you now, we started decentralizing the Ayurvedic hospitals the long time ago. Even the India, before India started having the Ayurvedic hospitals in decentralizing process. So we have a very well established Ayurvedic traditional system implemented in this country, and we are the only country who is having a separate ministry for the indigenous medicine. And more than this, we have this traditional knowledge. Quite a large number of traditional healers or doctors are practicing their medicine in this country. So also we have about 100 privately owned registered production centers, and there they use about 269 different medicinal plants in their production, maybe in dry form and fresh form. So we have few companies that do exports of medicinal plant-based products and medicinal plants to various parts of the world. But the main import of our medicinal plant is the European region or the European countries. So if you look at the global trend or the global market for these medicinal botanicals, as MRFR projects that the global herbal medicine market will be 111 billion by 2023 with a growth of 5.88%. And WHO at the same time, as you all know, the predicted 5 trillion market by 2050. For herbal-based products. And India is already a major exporter of medicinal plants to various developed nations of the world. And as I obtained from the literature, it is estimated that about Indian rupees 860 billion worth of raw materials and drugs obtained from these medicinal plants are exported from India. And also India is expected to be among the top 5 countries in the world with respect to the absolute growth in terms of the revenue from herbal sector. And if you see like the recently in 2017 and 18, during that time, the India exported about the medicinal herbs worth of US dollar 330 million, and the products as products, of course, about US dollar 456 million. So that's this graph shows you the trend, what we see from in next few years to come. And as you can see, for various products, the global herbal market is growing steadily, so that these herbal based products can be marketed with this back to nature concept that we see in this world now. And if you look at the phytochemicals as drugs and drug leads, as you all know, although the synthetic chemistry provides the basis for pharmaceuticals, for allopathic medicine, still about 50% of current drugs has a root to phytochemistry of medicinal plants and medicinal microbes. Now, according to Newman and Craig, there were 1,562 new drugs approved between 1981 to 2014. So out of those, about 45% had natural products as their source. And also, at least some of these, the developed drugs, has some leads from the traditional knowledge. So as you all know, in allopathic medicine, in the pharmacopoeias, the well-known drug morphine, which was discovered in 1800, also the aspirin, which discovered from willow tree, is still a function in the allopathic medicine as wonder drugs. And also the cancer drugs, such as the Taxol, vincristine, and vinblastine are yet to be the main chemotherapeutic agents for cancer, which are actually isolated from medicinal plants that are available in this part of the world. And we see the Pacific Eve, although it is found in US, we see that it is found in I think in India as well as in China. China, of course, I know for sure, because I have been to one of the Chinese medicinal plant gardens where you see the Taxus yunanensis is there in China. So if you look at the evolution of this phytochemistry-based medicine, that is the isolation, characterization, and coming as the allopathic medicines, it has evolved over the years. And if you see, like in the ancient time, the use of these medicinal plants are basically the experience based, and if you see, like the earliest documented record is from for plant-based medicine is from a grave of Neanderthal man from the archaeological site in Iraq, which dates back to about 60,000 BC. And although the scientists knew that the curative agents are there in these medicinal plants, but in the 17th and 18th century, the knowledge of phytochemicals grew rapidly. But in these 2 centuries, it was not able to uncover the active molecules from these herbs until we discover the morphine in early 1800. And that was the breakthrough that has actually taken place on isolation and characterization of active ingredients from medicinal plant. So the morphine was isolated from the opium poppy in 1817, and by 1826, the Bayer started the marketing of these natural products for pain-killing purpose. And you all know the most illicit drug is also out of this morphine, which is diacetyl morphine, the heroin. So that is also based. If you look at it, actually, it is also from a medicinal plant. So now in 2600 BC, as I told you, the earliest documentation of use of medicinal plants arose from ancient Mesopotamia. And then as I told you, the morphine isolation, characterization, and commercialization was taken place in 1827, and it became the first pure natural derived medicine. And with that, in 1820 to 1850, there was a boom in the isolation and characterization of various bioactive molecules, and discovery of some well-known natural compounds such as quinine, colchicine, codeine, salicine was taken place during this time. There was a small lag period, and then in 1929, the Fleming discovered penicillin, and he won the 1945 Nobel Prize for Physiology and Medicine. And then in 1952, the Nobel Prize, 1952 Nobel Prize for discovery of streptomycin induced the discovery of the bioactive molecules from plants to a great extent. And then in 1959, the first marine-derived anti-cancer drug was reported, but thereafter the phase of discoveries from natural products actually phased off to a certain extent due to the pharmaceutical companies were more focused on the synthetic chemistry and high throughput screening. But later it was found that the discovering from nature has more potential than discovering from the man-made chemistry libraries. And with the Nobel Prize in 2015 for physiology and medicine for Campbell and Omura for the discovery of ivermectin and artemisinin. So now there is a greater tendency of discovery of medicinal bioactive natural products from medicinal plants, as I explained to you in my previous slide. About 1,554 compounds that was approved between 1982 to 1914. 2014, I'm sorry, as from at least 45% are from natural products. For the interest of the audience, especially for the students, I have lined up the important compounds that was discovered from natural products or medicinal plants in chronological order, starting from the morphine and to salacinol, which is a compound that was discovered in 1997 from one of our Sri Lankan medicinal plant called Salacia reticulata. Of course, Salacia reticulata or the Salacia other species are found in India, but this discovery was made from the plant that is found in Sri Lanka called Salacia reticulata. So this is for the interest of students especially, and I have aligned this in a chronological order, and I'm not going to explain everything here. And the morphine story I told you, there is no drug as yet to replace morphine. So it was discovered in 1800. Now we are in the 21st century, but still this drug remains. It's a molecule that came from a medicinal plant, and there is no alternative has come as yet. But there is, as you all know, there is one compound called epibetidine, which is 200 times potent than this compound, which is isolated from the skin of the poisonous frog, but which has not actually has gone to even clinical trial level, as I understand. So this drug remains as it is for the last 200 close to 200 years. And also the story of aspirin, as you know, it is from another medicinal plant called the willow tree, which is actually discovered in 1899. So it has passed the 100 years. In few years back, we celebrated 100 years. One of the remarkable discovery from a medicinal plant. And I have aligned this in chronological order of the long way that this indigenous knowledge marched through over the years. And as we all know, in 400 BC, this was actually given for women as a tea to relieve from pain of childbirth. So it's went on various discoveries. The first clinical trial was done in 1897. And then this aspirin became a generic drug in 1930. And by now, as you all know, the aspirin is known to be useful in various other diseases, especially in the cardiovascular diseases, and to delay the onset of senile dementia. And aspirin is being tried for various other diseases, including diabetic, pregnancy complication, and coronary artery disease. So these are some of the drugs that are found in allopathic medicine, which has derived from the medicinal plants and medicinal microbes. And one other important area of when it comes to the diseases, the antibiotics. So as you all know, the antibiotics discovery is taking place, took place over the years, and you know, at the same time, the resistance is building up for all these antibiotics that we know. So we need another breakthrough or finding of new antibiotics from the nature. So world therefore is still relying on the phytochemistry for the next revolution of antibiotics to face these challenges. So this diagram shows you the discovery, the chronology of discovery of antibiotics. But at the same time, on the bottom, you will see how the resistance building up for all these antibiotics. So there's a big vacuum to be created in the years to come on antibiotics that will be able to resist these deadly disease-causing bacteria. So now in this antibiotic line, the two natural products originated from Sri Lankan microflora, namely lancomycin and lancacidine. Lancacidine is sold as sedamycin in Japan for swine dysentery. And these two drugs are trying for the deadly MRSA or methicillin-resistant staph disease or the infection by Professor Ada Yonata. So Professor Ada Yonata is the Nobel laureate in Israel who won the Nobel Prize in Chemistry in 2009. And she's the first Israeli woman to win the Nobel Prize out of 10 Israeli Nobel laureates, and the first woman to win in 45 years to win the Nobel Prize for Chemistry. And that's me long ago with Ada Yonata. And so she's trying these two Sri Lankan compounds for MRSA infections. And as you all know, the phytochemistry plays an important role in food plant as well. And the food plants and microbes provide the most needed phytochemistry to sustain the life, which science defines as macro to micronutrients. And the food plants containing phytochemistry with biological activities are identified as the health food or functional foods. And the fractions or purified fractions from those bioactive portion of the functional food are sold as nutraceuticals. So these are some of the vegetables or the edible plants in Sri Lanka. And if you name any of these, the traditional knowledge has some curative purpose for that particular plant. So what we eat almost every day, almost all these plants carries some sort of health benefits, which is recorded in our traditional knowledge. And at the same time, you know, we known as the land of spice, and all these spices has some health benefits for you to maintain a good health. So our food diversity is very rich, and our food diversity is almost all in that diversity are functional foods. And we also have a very organized system of living in the village setup, and we have aligned our living systems to a seasonal tank based arrangement, where you have the village tank and a cascade system. So basically these villages are self-sufficient, although now we don't see with this urbanization, these kind of villages they are operating as it was. But now the government is trying to reintroduce this with the organic cultivations and organic inputs for their cultivation practices, including the cultivation of medicinal plants. So we all know that our ancestors were very clever, and they have used the genetic resource, and they manipulated the genes using the knowledge on biodynamics and the environmental and climatic processes. And they, you know, they do vertical gene transfer knowingly or unknowingly through their conventional breeding for crop improvement, domestication, diversification, climatic adaptation, and so on. And that leads actually to the diversity of medicinal, diversity of the phytochemistry that we see in these plants. A good example is this Brassica juncea. So this was actually crossbred and keep on crossbreeding and resulted all these different vegetables that we see today. So as you see now, you don't see phenotypically no relevance to the original plant. And this domestication and vertical gene transfer has caused this diversification to the phenotypic diversification as well as chemical diversification that we see today. So there are some interesting examples that have validated this traditional knowledge through modern science. I myself have involved in several experiments to validate this traditional knowledge, especially some informations that are given in Susruta Samhita, the famous Indian text. And we have scientifically proved the claims that this Samhita has claimed. And of course, we know that certain factors are beyond the knowledge of modern science. So some we will not be able to prove until we have generated more scientific knowledge in the years to come. Now, specific health benefits of specific plant species is now proven to be due to the presence of these specific phytochemicals that can prevent or cure disease. So that is proven. And these phytochemicals are working even at a level of regulation of genes of an individual. That also we have seen. In fact, my own studies on sesame lignans on hepatic lipid metabolism has proved that the sesame oil on hepatic lipid metabolism have proved that specific phytochemicals in sesame oil, that is sesamin and episesamin, is able to regulate liver enzymes by regulating liver genes that are responsible for the hepatic fat metabolism. So the ability of phytochemicals working at gene level may have been known for our ancestors. We don't know, but I think they may have known about this. So this knowledge anyway have led to the traditional knowledge what we have now, that is the health foods, the art of healthy living, the ability of indigenous medicine in permanent cure of disease or a symptomatic cure, what we see in allopathic medicine, the probiotics, the prebiotics, the knowledge on correlation of gut microbiome to the body functions, the cosmetics and cosmeceuticals, the biocontrol of agricultural pests and diseases, and also in the organic or the nature farming. So our work on Sri Lankan anti-diabetic plant called Salacia reticulata, in Sinhala we call it Kotalahibutu. It showed all plant parts contain glucosidase inhibitory activity at varying levels. So we studied all plant parts of this well-known antidiabetic plant called Salacia reticulata. And we demonstrated that the alpha glucosylase inhibition according to the plant parts. As you can see, the IC50 here is higher in the leaf, and if it comes to stem and the root, IC50 is very low, which means it has more of the active ingredients than in the leaf, twigs, and the stem, the tender stems. Likewise, we have done the anti-glycation ability of these various plant parts, and again the same result. As you can see, in the stem and the root, you have more anti-glycation activity. The responsible two compounds are salicylic and cotonolol. So these two are the glucosidase inhibitors found in this plant. And this is a very interesting plant. As you know, use of this root of this plant in traditional formulations for diabetics is remarkable. Example, to explain the richness of our traditional knowledge, because scientific findings on this has evidence that this plant possesses a multitude of diabetic control mechanisms. So it is within the single plant, you have different mechanisms. We have observed different mechanisms of controlling diabetes. So Kotalimbutu extract or a decoction controls blood sugar by inhibiting starch breakdown enzymes in the gut, preventing the release of glucose from the starchy food, so that it can bring down the GI of any food that you eat. And it is also well proven now, Kotalimbutu extract control blood sugar by blocking the activity of enzymes, alpha glucosylase, beta glucosylase, and alpha amylase. And also Kotalimbutu has the ability to block gluconeogenesis in the liver, and by inhibiting the gene for gluconeogenesis rate determining enzymes in the liver. And interestingly, this remarkable plant is also able to control blood sugar by inhibiting conversion of glycogen to glucose in the liver. And also, as you all know, the diabetic complications are due to the formation of advanced glycated end products. So this plant has showed a remarkable ability to control or block the glycation process, as well as the same extract is able to reverse or break the already from glycated end products. So this is for the first time we demonstrated, and we published this in one of your book published by NAM SNT Center in India. And these are all the diabetic complications that I have put here are due to the formation of advanced glycated end products. So controlling of advanced glycated end products is important in diabetic controlling diabetic complications, and reversing, if possible, is very important. And based on these findings, our team at Industrial Technology Institute developed a low GI biscuit using the glucosidase rich fraction of this plant. And now this biscuit here, as you see, this biscuit is now in the international market, and it is the lowest, the biscuit that carries the lowest GI in the world. And this is called Kotalimbutu biscuit, and it is now available in some of these overseas countries, which is exported from Sri Lanka by one of the leading biscuit manufacturers in the country. And we also worked on the phytochemistry of the marine algae. Just to complete this talk, I thought of putting some of our work on marine plant as well. And we isolated the world 8th non-human sperm motility stimulant, which we code name as XAT1, which is 28 times potent than the current clinical human motility stimulant pentoxifylline. And from the same algae, from the same fraction, we also discovered a non-steroidal antiprogesterone compound, we called name SPC1, which can be developed into a once-a-month pill that can prevent unwanted pregnancies. This is the exact one, and this is the SPC1. And we also worked on the unknown aromatic plants. Of course, we have a quite unique mountain rainforest, and we explored this and we discovered some interesting perfumery and compounds that are useful in perfumery and culinary industries, as well as in aromatherapy industry. And as you know, the elemicine is, elemi oil is a very famous aromatherapy oil. And we came across a plant with which is rich in elemicine in that oil. And we also studied the Ceylon cinnamon extensively and conducted a clinical trial, and it passed its phase 3 clinical trial recently. And we are in the process of developing this nutraceutical out of this cinnamon, and will be available in few months to come in the market for treatment of diabetic. And this clinical trial showed that this extract from this particular cinnamon species, it has anti-diabetic and anti-lipidemic activities, which has a very strong nutraceutical potential. We have a few small groups actually operating in the bioactive natural products. This is the Colombo University group, and these two professors are working at the Colombo Medical Faculty. They are mainly interested in the anti-diabetic plants, whereas this team is mainly working on marine natural products, where I was at the beginning was involved in this group. And then later on, I became interested in both medicinal and aromatic plants when I was working at the CSIR Sri Lanka. And this is the largest group in Peradeniya University. And you may be knowing some of these senior colleagues of you all. And of course, Professor Vijay Kumara is from India. He actually from Kerala. He migrated to Sri Lanka and he's living here. And he's one of the leading natural product chemists in the country. And CSIR Sri Lanka is, of course, mainly worked on essential oil and medicinal herbs. And quite a considerable work has been done by these leading scientists at CSIR. So I moved to CSIR immediate after my PhD and worked on the medicinal and aromatic plants until I moved back to the university. So we must be knowing R.O.B. Vijaysekara is a famous natural product chemist in the world who worked here in his early career, and he moved to UN, and he's a famous scientist in the aromatic plant area. And we also data mine all Sri Lankan natural products published from medicinal plants during the last 2 decades. And we screened 480 different compounds in silico for several potential drug leads. And we have come across very interesting compounds for ACE inhibitory activity, aldose reductase inhibition, and renin inhibition. And interestingly, when we look at these compounds and we retrospectively, when we look at these compounds to see from what type of plants these are originated, those who actually originates from medicinal plants that are given in our traditional knowledge, traditional medical systems for hypertension, dyslipidemia, and cardiovascular problems. So it is quite interesting when you look at retrospectively, they correlate with this traditional knowledge that we have. And at the same time, with this COVID pandemic, we thought of screening these compounds, the 480 compounds for COVID-19, the main drug target, the enzyme, main protease enzyme MPRO, and which is responsible for mediating the viral replication and transcription. So we are very happy to disclose that we got 3 compounds with strong binding energy for above key enzyme MPRO. So we are working on these 3 compounds for potential application in COVID-19 treatment. So in essence, the phytochemistry of medicinal herbs will continue to be the source for many interesting compounds for biomedical, agricultural, and industrial use over and above of any sources, including synthetic and man-made compound databases or repositories. Thank you very much for listening to my speech. Thank you very much.
Thank you, sir, for sharing valuable information about the diverse flora of Sri Lanka and their medicinal importance. The details shared by you would be really helpful for the budding scientists and researchers in the field of pharmacognosy, botany, and life sciences as well. So thank you very much, sir, for the detailed information and very informative session.
Thank you
Sir,
very much. I have given my email and I would like to keep in touch with all of you. If you are happen to come to Sri Lanka, please drop a mail before you come. I will take you all around these two interesting institutes in this country. Thank you very much and give my regards to Professor Mamta, and I am thankful to her for inviting me for this important conference conducted by these two nice ladies here. I can see you. Thank you very much.
Thank you, sir. Ma'am, Mamta, ma'am.
Hi, thank you all. Yeah, Dr. Premal, Prema Kumara, thank you so
Yeah,
much. Yeah, thank you so much for giving such a nice presentation. I shall hand over the mic to Dr. Preeti now.
sir, can we have the question and answer session at the end of both the sessions? Will that be convenient to you?
yeah. What time you will be closing the session?
At around 1 p.m., sir.
Okay, right. Yeah, no problem. I'll stay.
You'll be here, na, sir?
Yeah, I'll be here. I'll stay.
So
Yeah.
do please
Okay.
stay with us then. Yeah.
Okay. So participants will be having the question and answer session at the end of both the sessions that will be conducted by Dr. Mamta Shah. Our speaker of today's next session, Dr. Shankar Katai, has joined us. He is going to deliver a talk on propolis for holistic health and state of botanicals in the UK. Let me give you a brief introduction of Dr. Katai. He is presently a quality and research manager in Nature's Laboratory at Whitby, UK. He graduated from the Institute of Pharmaceutical Education and Research, Wardha, India. Thereafter, he pursued MS Pharmacy in Natural Products at Naipur, Mohali. He has also done postgraduate diploma in Chemical Technology Management as well as PhD in Pharmacognosy and Phytochemistry from ICT Mumbai, and also holds a degree of postdoctorate from University of Bradford, UK. He has around 10 years of experience in the field of natural products research and development. He has served as Quality Development Manager, a Knowledge Transfer Partnership Associate at University of Bradford, United Kingdom for 2 years, and as Research and Development Scientist at Netrion Inclusive, Kolkata for 1 year. He has also contributed to the academics field as an assistant professor at School of Biological Sciences and Biotechnology, Institute of Advanced Research, Gandhinagar, and also at Department of Natural Products, Naipur, Ahmedabad as well. He has contributed as a senior research fellow in an ICMR project in which he was involved in development of 30 monographs that are now published in 11th to 13th volumes of quality standards for Indian medicinal plants. Dr. Katai is a recipient of several fellowships, including Innovate UK KTP Associate by Government of UK and C.V. Raman International Fellowship for Indo-African Students, DST, from PICI, to name a few. He has received various certifications and awards on ISO 9001-2015 Internal Auditor from Advisora, on IPR from IP Masterclass, Government of UK, and many more. Till date, Sir has published 27 research and review articles in various peer-reviewed national and international journals, and also has 8 books and book chapters to his credit. He is a dynamic member of Phytochemical Society of Europe and other important bodies as well, and serves as reviewer and editorial board member to various journals of repute. His research area of interest includes identifying correlation of chemical composition and biological activity of propolis from all over the world using multivariate correlation analysis. He holds an expertise in analytical method development and validation, quality management, ISO 9001-2015 internal audits, management representative for ISO and organic certification, in-house process enhancement, communication with various university and industry collaborators and partners. We feel obliged to have you here today, Dr. Katai, so we extend a hearty welcome to you. And now I request Dr. Katai to please start the session. Over to you, sir. Sir, your mic is mute. Yeah.
Hi, good morning, good afternoon to you all. It's morning here, 7:30. And in the beginning, I have a kid in house. It's 7:30 morning. So there may be, few interruptions. So please forgive me for that. And yeah, so should I start sharing the screen? Yeah. Is it visible?
Yes, sir.
Okay. So what I understood, the participants are they graduates, masters, are they pharmacy in general, or are they pharmacognosy master students, or how is the audience? So that will help me to
So there are graduates, postgraduates, and faculty members also. They are from pharmacognosy, phytochemistry, botany, and life science departments.
okay. So throughout the country. So
Yes, from
oh,
India, throughout the country.
all right, throughout the India. Yeah. So thank you very much, Professor Mamta Shah, for inviting me on this international conference. And I would like to speak about the propolis in general. So that's a very niche area, I think. And so to broaden and to benefit the students, I included about the general status of affairs in UK about the herbals. So presentation is basically in two parts. So before moving to very narrow topic, propolis, as we all know, the honeybees produced various products which we use, starting from very important honey, the beeswax, we all know the bee venom for many therapies, and the pollen, bee pollen, and the royal jelly. So, and amongst them is the propolis, which is not very well known in India as far as the graduate and postgraduate level and research level is concerned. So again, the beekeepers, they are not very aware about the propolis, its properties, its importance, and how they can benefit the economically from the propolis. So yeah, this is about broadly about the honeybees, various products. So there is a bit of history to propolis. Propolis is a natural resin collected by bees from the plants. So various parts of the plant and the birds and the exudates from the plant. They mix it with the wax and blend it with their enzymes and use it for their own defense. So it's like a natural immunity to them, or a natural antibiotic to the beehive to protect them. So if we go back into the history, so 5000, 5500 BC, the ancient Egyptians used it to mummify, to use as a resin to bind the protect the mummy. So that's the the ancient importance of propolis, because it has got a lot of antibacterial properties and various other biological activities, and very potent. So if you go, the propolis term was coined by Aristotle Alexander. Um, so pro is like a before, and polis is a city. So protector of a city, a defender of a city, which you can correlate as a defender of the beehive, which prevents the inside larvae and the queen bee and other bees from the external environment, and produces the sterile environment, maintains the sterile environment inside the beehive. So that's the it's like a defender of a city, propolis. So coming very close, like 100 BC, the Roman soldiers used to carry it with them when they go to war to take care of any wounds and injuries. And women used to consider it as a friend for the propolis uses for various purposes in the form of propolis cream. So that's the brief history. In 1600s, propolis had found its place in London pharmacopoeias at that time. So, and in 1940s, when it's like during that time and after that time till World War II, the Russians used to carry this with them and use it for healing the wound infection and for speedy healing. And they used to call it as a Russian penicillin. So that's the importance they knew that time and its potential for and wound healing properties of a propolis. And the Dr. Carl Agard, a Danish biologist who earned a nickname as Dr. Propolis for curing the 50,000 Scandinavian patients, which were not able to cure with other therapies that time, and it's like without any side effect. So that's um, the importance of a propolis in Scandinavian countries like Finland, Denmark, and Netherlands, that side. And mainly the Mediterranean region, Mediterranean countries, the propolis is very, very famous and still very, in a very much use for taking care of the wounds and the diabetic complicated complications related to wounds, and which are not able to, um, cure or heal properly with the normal the modern days medicine. And in 1990s people started up talking about the benefit of propolis, and for the ailments and the areas health problems which are not responding to the modern medicine. So from 1990s, truly the propolis has got kind of attention by the scientific community in terms of exploring its properties, studying it with the scientific way, and verifying its ancient claimed activities. So general practitioners started prescribing these to the patients from 1990s in European countries and the Mediterranean countries. So yeah, after about the brief history of propolis, so that tells us about the how propolis is very potent and has got a lot of biological activities. So Greek and Roman physicians used it for mouth disinfection, antiseptic, healing properties for wound, as I said, for cutaneous therapies, any allergies, and psoriasis and various other skin-related problems. So propolis has got very good applications on skin-related problems. So amongst the plethora of biological activities, in our research, we focused on antibacterial and antioxidant. So because they are very important, although it has very, it's like a broad spectrum of activities, and you can say the holistic spectrum as bees collect chemicals from the plants and various plants, and so plant, if the plant has an activity like anti-diabetic or anti-tumor or anti-inflammatory, then those chemicals are going to be there in the propolis, and ultimately propolis will be responsible for that activity. So that's the benefit of propolis collected by bees. So when we were doing research and searching literature for our studies, I came to know that throughout the world there are various professors who made their whole career researching the propolis, but in India I found very few. There are scattered like one odd articles from different groups, but to mention prominently, Dr. Anil Gupta, most of you must have heard about him. I think retired in 2017 from IIM Ahmedabad. He successfully ran the grassroots innovation and honeybee network that basically give information to the beekeepers how to do beekeeping and the importance of various products of honeybee, and particularly the propolis. Last year, I think 2018, I visited CBRTI in Pune. So they have very good facility. They train the beekeepers. They tell them how to take care of bees and how to cultivate the propolis and the importance of propolis. In my opinion, propolis is like gold. If you see the price, it's like a gold resin. But unfortunately, not many of the beekeepers know that black sticky substance on their honey collection frames. What's that? And they just throw it. So that's the sad part. Professor Mahadik and Professor Anand Paradkar from University of Bradford, they in collaboration have been publishing from many years now, maybe more than 10 years on propolis. So there was a few publication from Deepak Kasute, Dr. Kasute. I think he is in Bharati Vidyapith. So apart from them, there are many other researchers in India who did research on propolis, but very, very limited publication. So it's not very vastly famous topic, and it's very promising. I mean, it can be taken up as a research projects or research in propolis by scientists in India, because as we know, the bees collect these chemicals from plant, and India has got diverse flora in Western Ghats and the Himalayas and in various parts. So our climatic and tropical environment makes the plant flora very diverse and has got very prominent biological activity. So the properties of propolis in India would be much more diverse and potent for various diseases prevalent in India. So this can be a very good candidate for research activities. These I have just put some pictures of propolis we collected from different countries, and as you can see a very vast difference in the texture, brittle, color, and the taste of the propolis. So based on the geological flora and fauna of the place, the kind of regions bees are collecting, the nature and texture of the propolis changes. So that just for your interest, I put those pictures here. So coming to how bees process the how they produce the propolis. So basically, when they have a slight attack on their colony, or any stress, infection, or any rodent or outsider coming into their colony and trying to infect, or maybe if there are cracks in their the honeycomb, then what they goes out foraging the resins from the trees and plants, and they mix it with the wax from the wax glands and the enzymes, and that basically makes a very sticky resin, and they put it on the top of the hives, and that we collect as a propolis. So that's basically the how bees process the propolis. Here you see bees is collecting the propolis from and putting it on their hind leg, bringing it to the frame, and you see the black trap with the propolis, full of propolis. So when the stack of the propolis collection frames are kept on the top of it, generally the black traps are kept with the small gaps. So what the honeybees do is like they try to seal that gaps and to protect the colony inside. But we can take out that trap and take out the heavy load of propolis. That's how propolis is collected, on commercial scale, because propolis bees doesn't produce propolis unless they need it. So we are creating a need by putting the porous mesh over the honeybee collection trap, the frames. So that's how commercially the propolis is collected in huge quantities. Sorry, you may hear that interruption in between. So yeah, so moving towards the chemical composition, it has basically 50% resin, which is more of our interest in this, 30% waxes and 10% essential oils and 5% pollen. So out of that, so till now, with the advancement of analytical techniques like HPLC, LCMS, GCMS, and other techniques, more than 300 chemicals are now have been identified from the propolis throughout the world. And these are very few commonly observed chemicals in the propolis, which we categorized as a flavonoids and phenolics. This is basically for our study, we used two class of chemicals. So these are very commonly, the most important amongst the flavonoid is galangin and pinosembrin. And amongst the phenolics is caffeic acid phenyl ester, which we call as a CAPE. So basically these principles contribute to the activity of a propolis, and amongst others. Yeah, so when we were doing a research, we thought the temperature zones in a world will has an effect on the plants growing in that region. So to see what kind of differences are there in chemical compositions of propolis collected from these three regions. So you can see the red zone, which is like tropical, hot and temperate region, hot region, and subtropical, which is like Mediterranean, in between two temperate and tropical regions. So we used these map for making a classification for analysis of the propolis from these regions for our study. So, and then we analyzed propolis, which was taken from various countries like Brazil, Finland, Africa, the Europe, again from America, Canada. So from various places we got propolis and analyzed it by HPLC to see what are the differences. We found that the propolis from Mediterranean region, particularly Turkey, Libya, was very high in the number of chemicals present in them. So this is just a representation of a propolis analysis with the 13 markers which we selected for our study. We selected those chemicals for performing a multivariate correlation analysis by using a statistical tool, SPSS. So there are various other tools available for analysis for performing these correlation studies. So what's a propolis bioactive quotient? So when we say chemical composition of a propolis will have effect on its biological properties. So what's the correlation between chemical and propolis bioactive? So effect of chemical composition in a propolis and its effect on a biological activity. So we basically took a range of chemicals, as I showed, flavonoids and phenols, their total flavonoid and phenols, and their effect on biological activity like antibacterial and antioxidant. So by using regression analysis, we performed the study. So it's like phenols, flavonoids, and the individual 13 chemicals were our independent parameters because the concentration sorry, the dependent parameters were obviously like the biological activities, antimicrobial and antioxidant, which were dependent on the chemical present in the propolis. So while doing this, we found that there were like lack of lot of challenges in pulling the data from these three the environmental zones. So when we actually searched the literature, we found that there was not uniformity in the reporting the biological activity. Like sometimes there was a MIC reported, sometimes there was like the zone of inhibition was. So there is a difficulty to correlate between them, and sometimes like sometimes few groups used different bacteria. So when you are performing a multivariate correlation analysis, you need good number of valid cases or studies to be incorporated. So you cannot incorporate one study performed on E. coli or another study performed on Bacillus subtilis or any other bacteria. So you need to have common bacteria, common output like MIC or maybe zone of inhibition. And then again, the geographical and seasonal variation was the factor which is common in the samples, which made a lot of variations. And the limitation of analytical techniques, because if you are selecting a study where the instrument was HPLC for reporting chemical composition, then the level of flavonoids will be higher than compared to the publication where the GCMS was used. So their flavonoid content is bound to be less, and more of fatty acids and volatile content was more reported. So correlating, different chromatographic techniques, research studies was difficult. So we had to, leave a lot of studies which were published to be considered in our model. And we published a small review gaps in propolis research and challenges for the commercialization. And then through that study, we came up with the propolis bioactive quotient for the classification of a propolis, because at that time we did not have any kind of classification how to grade the propolis. Is it good, active, or inactive? So after putting all the parameters inside the statistical tool and using the model, we came to know that how many milligram or microgram of how many milligram or microgram of propolis is required to show IC50 for antioxidant activity, or maybe the MIC for antimicrobial activity. If you put in our model, which we developed for designing a PBQ, then that tells us the propolis is very active against has a good potential as an antioxidant, or has a good potential as antimicrobial. So you can grade and classify the propolis based on this model. So that was a study we performed. So final outcome, as we all know, in a tropical environment, so a lot of hot, humid weather, so conducive for growth of microbes and bacteria. So the plants will have that stress on them, so they will synthesize those chemicals, and the bees also living in that environment will have a stress of bacteria. Just a moment, sorry. Shambhavi. So sorry, the bees also living in the same environment. They also have a higher load of bacteria, in the colonies. So they go and scavenge for those chemicals with antimicrobial potential. And then in our study, what we found, like in tropical propolis, the phenolic contents like capic acid, cinnamic acids, the benzoic acids, the K was higher in concentration through our HPLC studies and through our data analysis from the earlier published publications. So we found that these chemicals contribute to the antimicrobial potential. And looking at the temperate region, which is like colder European countries, or the Russia, or the Siberia, or the Canada, and the northern part of China, where it's like cold weather is prevalent, then that we call a polar or subpolar regions and temperate regions. So there it's like inflammation is higher. That's a factor. So they doesn't have a lot of microbial load on them. So plants manufacture a lot of flavonoids. So what we found that the propolis from temperate region had a high amount of flavonoids, and that contributes to the antioxidant activity. We all know that flavonoids have a very potent antioxidant activity. And when we saw a subtropical, so middle band in between two regions, these tropical and temperate, we got intermediate phenolic and flavonoid content and intermediate antioxidant and antimicrobial activities. This is basically statistical tool applied outcome by using multivariate analysis on SPSS software. So yeah, that's the outcome of our study about the propolis. And I thought speaking a little bit about the propolis and COVID correlation has propolis has any kind of applications in this situation. So propolis is very well known for its activity against the herpes zoster viruses and influenza virus. So that studies are there if you want to check. So, but the direct study against the COVID-19 is not, obviously it's being a new novel virus. But the a group in IIT Delhi and some group in Japan, they have done some computational studies on vilanulides and the chemicals from the propolis. So they found this caffeic acid phenethyl ester, which is CAPE, very important molecule in propolis, is having a good affinity towards the receptor of a COVID protein against the protease. So that was like computational based, need to be proved with the actual lab testing. That's one study. Another study by the Bakenski Dimitri. She's a very big scientist, and I think the ISO wants to establish the international standards for analysis of a propolis. So she is the chairman of that committee who is designing the international standards for propolis. So under her watch, there is one clinical trial going on a Brazilian green propolis, because Brazilian propolis is considered as a gold standard amongst the various types of propolis throughout the world. So I think 120 participants are there, and you can see the link if you want to see the progress of this clinical trial. I guess it was when I took this reference, it was in clinical trial stage 2. So it's basically two references I found about the propolis. And as I said, propolis is immunomodulator, as a adaptogen, as an antibacterial, anti-inflammatory. And we know that in COVID, the interleukin storm comes. So if anti-inflammatory and antioxidant property of a propolis would somehow help to, prevent or reduce the interleukin storm. And ultimately it could be, of a preliminary or a prophylactic kind of treatment if you want to, keep as Ayush has recommended taking different kadas of using aromatic herbs. Likewise, the propolis can be used as a prophylactic so that your immunity stays in good shape. If you unfortunately, if you catch the virus, then it gives a good chance of your body to fight. So although the claims are not established, the studies are not finished, but looking into the chemicals and the other knowledge, this can be a good candidate for exploring its effect on COVID-19 and maintaining as prophylactic before before you get, to help you fight if you get it. So yeah, moving towards the next section of what state of botanicals in UK. This is basically information I collected from various government sites. It's not my research area. And as Professor Mamta said, that a lot of students will be their graduates, so keep it a bit general, also not very specific towards research or propolis being a niche area. So yeah, so quickly I would let you know what kind of status botanicals have here, what kind of government norms and guidance are there. So very early in 1538, the William Turner has written his work described as a British fauna and fauna. And now the British Society for Britain and the Botanical Society for Britain and Ireland takes care of this, botany related research, identifying new species and documenting them and other activities. So moving towards, I found while I was searching, I found a very interesting survey by Professor Michael Henrik from University College of London. And they basically ask people, how do you think about the herbal medicine? Do you take it? And how their perceptions is about the herbs. So they found that age group 30 to 55 particularly is more popular amongst them, and 68% of indicates 60% of the participants indicated that plants are effective, and because they are natural. And the around third of the people grow their own plants, basically the teas, herbal trees, the rosemary and lemon balm, they are quite famous here rather than medicinal plants. The culinary and the aromatic plants and the teas, they are specifically more famous in UK. They also found that the participants hesitate to tell their the doctors, physicians, and the general practitioners about what kind of herbs they are consuming, as a normal health maintaining food supplement. So that's the worry they found, and it's widespread application of these products. They suggested that the high quality standards need to be set, and this need to be prioritized by the government. So this was a quick survey, and that tells us about the awareness about the product in the general population and state of where the information they get their information about this product, how they think this are they safe or so yeah. Moving towards the next point, the Medicine and Herbal Healthcare Product Regulatory Agency generally manages all the medicine related aspects here. So on their website, you can see the list of entries or herbs they identified as a risky herbs, or either they are prohibited or they may be limited. Their use is limited and controlled. So there are about 62 entries throughout the different plants, but I selected just plants which are very known in India, common in India. So I excluded others. So this about tells you the list of, and these herbs are basically poisonous herbs or maybe very potent herbs, or herbs with having interactions with other modern medicines by, affecting the enzyme metabolism of antibiotics and the modern medicines. So basically it is, understood that these herbs are high risk and need not be easily available to common public and has to be either given to the patient by pharmacist or registered pharmacist, or under the supervision of pharmacist or a registered doctor under their prescription. So basically list I highlighted one Areca catechu. So we generally consume it every day, every household in different form of pan masalas and pan supari. So that has like carcinogenic potential, and this country says that's limited, and we in India very widely use it every day. So there is a kind of list you can I think you all know about these herbs. These are very high potent or maybe poisonous or having interactions with other medicine. So just a list I just wanted you to go through. So this comprehensive list and the dosing is easily available on the website, where the doctors and the common public can definitely see they are using this herb, what's the dose and what's the like legislation, are they restricted, are they poisonous, something. So that's why I wanted to show like very precise, concise information available on one place, the link we provided there. So that kind of very informative information needs to be there in India also, I think. I'm not sure whether that's available on Ayush or ICMR website or any other website, government site. So yeah, moving towards another important topic in EU. Very many years back, there was like a huge debate about pyrrolidine presence in the food coming from the plants, and they started investigation at Shambhavi. Sorry, kid running out of patience. Yeah, so these pyrrolidine alkaloids, they are basically carcinogenic. They have genotoxicity and they affect the liver, and particularly 1,2 unsaturated pyrrolidine alkaloids. So what they found that the borage and compresse species of a plant has got high level of these PAs, and these contaminates your food. They have selected many samples of honey and bee products, the meat products, and the teas, herbal teas basically. So they found that if the bee is foraging on a plant which has got high PAs like borage and camphor, then the honey is bound to have more of those chemicals. Likewise, if the teas is cultivated and has got some weeds which has got PAs in them among those 6,000 plant species, then the teas will have higher amount of PAs. And if the animal who is grazing on a weed or a grass or in the field, if that field has got a lot of plants with high PAs, then it will come into meat or a milk. So that's the source of contamination. And then they started awareness and analyzing different samples and identifying which plants are having high number of PAs. So their studies, they implemented some restrictions over the use of some herbs, and I think in 2014 they analyzed various thousands of samples, and they found out that the frequency and the amount of PAs available present in the normal food items have been reduced significantly. So it's very stringent norms they have for these PAs to be present in food and medicinal plants. Our company stopped using stocking these herbs and making any products by where we had borage and camphor. Again, there are few herbs like kawa, and they have concerns about. So these are the few herbs with high PAs, and they are very particular about this, not these chemicals exceeding the permitted level, as they are carcinogenic, genotoxic, and damages the liver. Third, as we have system in India, government hospitals, likewise here National Health Service, you all must have heard about it. So I thought, what NHS, because that's the only system through which you can go to the practitioners and get yourself treated. There are no or very less, I guess, private hospitals. So this is the only source you have. You assess your health facility. So I thought what they think about the herbal medicine in UK, because they are mainly modern medicine based hospitals giving any treatment, and the practitioners taking it when they're doing their practices, they study modern medicine, and not they don't have much of information about the herbal medicine. So this is the link if you want to read in a detail, and they think like the botanicals are natural, but they are not necessarily safe as you think. So potential issues, they found that it may have, and this is we also know that herbs has got some effect on the cytochrome enzyme system, so that they will affect the bioavailability of the and pharmacokinetic of the modern medicine. So that may alter their metabolism in a, it's like induction of an enzyme or inhibition of an enzyme. So their concentration may be exceeding the what we think, and or maybe sometimes they are the concentration of that modern medicine reaching blood or its target site is less. So that's understood, and that's why they say it's like it may interact with other medicine. It may have, the herbals may have their own side effects. They may not be from the regulated market or manufacturer. So these are concerns about the herbal medicines highlighted on the NHS side. When do you should avoid, and this is very common and applies everywhere, I guess, throughout the world, that when you are taking other medicines, when you have liver disease, because the metabolism will be altered of these herbs and other modern medicines. When you go to surgery, if some of the herbs affect your enzymes, which metabolizes the blood thinners or blood clotting agents, drugs, then the clotting time may get affected, and during surgery the patient may find very difficult situation. Complications can arise. Again, if someone is pregnant or breastfeeding woman and elderly and the children, so in these cases herbal medicine should be avoided. That's how the NHS says. Again, I said about surgery, what to look for while buying herbal medicines. They recommend like buy from the registered THR. So that's a traditional herbal register. So buy it from them. So all the herbs may not be having an evidence, so they may be having this traditional use. So look for the good quality suppliers with having THR registration. So I will speak about what is THR in the later slides. So risk of, now this is a trend online, everything is available. You find on Amazon and Flipkart, the others, eBays, the herbal medicine small packs are available. So that's very risky, because they may not be licensed from the licensed person. They may be coming out of UK unregulated market, or maybe they're not having a license to sell those medicinal plants, or maybe they have a bogus doctor or pharmacist on their sites, shown on their sites. They may be toxic substance because there is no regulation. So buying these medicinal herbs on online or mail is very risky. That's how they identified, and I suppose it is true, because not all the common population have information about the possible interactions with other herbs and chances of getting inferiated or adulterated or contaminated herbs. So if you're not expert in identifying the herbs, it's very risky to order these herbs online. This I found very fascinating when I was this is about the yellow card scheme, which is run by the Medicine and Healthcare Regulatory Agency. So what you can do is, when you use herb, have identified some adverse effect or complications with that herb, you can straight away register yourself and write it there, your story, and then information goes to the highest authority who makes regulations and the recommendations prohibiting some herbs or something. So that has happened with the St. John's wort, because it interacts with the cytochrome P4 system, P40 system, and affects the metabolism of a lot of drugs. That's why that's how they came to know about St. John's wort. And about the presence of heavy metals in Ayurvedic and traditional Chinese medicine. So this is how they came to know, through this yellow card system. It's a direct system to report the problems related with the herbs. So that's available on NHS. I found this is very important, because as the daily revolving situation, any new interaction or a problem herb can be real time monitored and reported. So this is, I thought, will interest you. So traditional so THR is basically a mark that gives you the confidence that the medicine which is produced and marketed in UK is of quality, because you cannot put these medicinal herbs on the counter, over the counter. They has to be bought through the medicine or a raw herb through the practitioner, registered practitioner, herbalist, or any registered pharmacist. So you cannot buy it open sources like from the on the shelf. So when someone is making a claim about the medicine, and again in UK, no one can make a claim about the biological benefits of any herb, medicinal herb. So if they are making, then that's the breach of law. Breach of law. So for that, making a minor medicinal claims, you need to have the traditional herbal registration. They give you mark, and then you can use it for marketing purpose, showing that gives a confidence about the safety and quality, because based on traditional uses, you can claim like for some pain related, cold related, cough related. So if someone is claiming a very significant major health concern like cancer or diabetes or hypertension or something, then it needs to go to the marketing authorization, where basically they check all the your studies. You need to prove by studying and doing research that this herb is effective against the any health condition. So basically it needs a bit of thorough study. That's traditional herbal registration. So without this, you cannot put a herb, sell your herb to the common man. And otherwise, if you are having this registration, then you can still sell the medicinal herbs, but through the practitioners only. So this is the link if you want to read more about this. Then coming to the last part, herb mark. So very recently, the British Herbal Manufacturing Association is moving towards regulating the manufacturing sector in UK, particularly for herbal medicines, because it's not regulated now how people manufacture and how they are there to the quality. And somehow government is moving towards putting more stringent norms requirements for manufacturing, those manufacturers. It's like asking for a stringent good manufacturing practices, which will be devastating. So what they are doing, they are preparing themselves through this manufacturers association to implement some it's like standards. Some you say ISO or maybe the British requires some quality system to be put in place. So the British Herbal Manufacturing Association came up with their own standard. It's the amalgamation of pharmaceutical and the food industry standard, so that it works best for herbals, as they are sometimes called food supplements in UK, unless you are making a claim that goes in herbal medicine section. So it basically has 9 aspects to be seen. Is basically want to make sure that when you are manufacturing a medicinal herbal medicine, you should know from where you're getting your raw material. You should know identity. You should have specifications. You should have system. In a production, you should have proper procedures, the worst instructions, so that when you produce, each time you produce a consistent product, you should have a properly trained person, properly suited premises and equipments, so that the kind of cross contamination doesn't happen and the quality product can be made. You need to have a documentation to show the diligence, because if something goes wrong and if you're recalling the product, you should be able to trace down from the raw material batch to the batch of finished product you send to the customer, and identify each customer you send that batch of a product. How system in a place like internal audits, identifying the critical hazards which can happen in your process. Sorry, Shambhavi, 5 minutes. Sorry. So in a production, you should have proper reported procedures and work instructions and specifications. You should ensure that the raw material coming in and the finished product going out is as per the specifications decided by you. You should monitor. You should be able to monitor the outsourced activities. If you are getting some of the products manufactured, are they adhering their products? Are they adhering to the quality standards? What kind of quality standard system they have, like ISO or BRC or GMP, GLP? What kind of system they have in their facility from where you are getting your product manufactured? How you should have a system to let complaints to be reported in a form, so that if the some of the complaints are about quality, then you should be able to find out the problems through your documentation system. And if there is any like a recall of a product, then you should be able to contact all the customer you send that batch. So, and self inspection and continuous monitoring and improvement. So basically, if it wants you to know what you are doing, if you are doing it today, you should be able to produce same product next time and again and again. So it should not happen like randomly someone thought, oh, let's make a herbal medicine, and just open some in a small facility, open some with some basic instruments, not having any system in place, started manufacturing and selling, and don't know from where he's getting, with what kind of quality material he's getting, and what kind of product he's making, and what to whom he's selling. You just know that should not happen. So there should be check and track of everything to ensure that the quality is maintained and the customer gets best quality product, and knowing the risk that this is a medicine, although it's herbal, it's a medicine, and someone's life is would be at risk. That may be fatal, or maybe small allergy, or small or major as bad as the fatal. So this herb mark, very recently they started this activity. They come to our facility. We have ISO 9001 and 2015 in place. So they observed our system, understood, learned, and again visited other manufacturing units who manufacture herbal medicine, and understand what kind of constraint, problems, and system we have. and to come up with the best solution so that we are not forced to the pharmaceutical standard stringent norms, plus we are not completely unregulated, so that any mischievous person can manufacture and put the life of common people in a risk. So that's the quality standard here. The BHMA is trying to implement. Yeah, that's about the state of regulations, understanding about herbs in people's mind, and the what NHS think about herbal medicine in UK. I think that's it from my side. Hi. Yeah. So if you have any questions, so I would be.
Dr. Katai, please stay with us for the question answer. It will take few minutes.
Okay.
Dr. Preeti.
Thank you. Yes, ma'am. Thank you, Dr. Katai, for sharing an extensive information on propolis and how it can be useful for achieving and maintaining the holistic health, especially its potential role in COVID situation. Thank you very much, sir. Participants, please note that the link for feedback form has been shared in the chat box. Make sure you write your correct names in that. Kindly fill it before 1:30 p.m. today, and you will be receiving your e-certificates on your registered email ID within a week. Now I invite Dr. Mamta Shah ma'am to please conduct the question and answer session for both of the talks of today. Over to you, ma'am.
Thank you, Dr. Preeti. I would like to invite Dr. Kumara. If he is there, we should be taking up the questions for him first. Sir, there is a question from Dharti Mehania. What is the main chemical present in morphine and how much amount in it is? Actually, she has wrongly written. Maybe what is the main chemical present in morphine? She has written. Maybe she wanted to write opium. And what is the amount of this compound in the opium?
Yes, I think it's coming from the latex. So the main composition of opium is in the latex. The percentage wise, exactly, I am not in a position to tell you what concentrations, but it is obtained from this opium, and I think from the literature you will be able to find the exact composition. I have not actually worked on this opium plant because we don't have that here, but I brought this in this discussion because it is one of the hits in the history of natural products, bioactive natural products. So.
Yeah, Dharti, I would like to add into this. Sorry, you have wrongly written over here. Morphine is a component of opium. It is a major component, and from that, many other components are derived like codeine, thebaine, and others. Right? It is a very valued compound, and it is used for the treatment. I mean, as a potent analgesic for the terminal patients of cancer. Right?
Yeah.
The next question is due to such a high demand and supply of the medicinally important phytochemicals, has there been a resistance in the land use when complemented with the typical natural farming and culturing? Also, like people have inclined more towards the growing flora, having rich phytochemicals and affected the per annum yield, and whether this has affected the per annum yield of the major crops. This is a question from Mr. Modan Maharun.
Well, in Sri Lanka, of course, there is no restriction as such, but the government is actually promoting the cultivation of these medicinal plants. Now, in fact, now recently, the government has declared a separate area through a gazette for organic cultivations. And there are also, the government plans are there to cultivate medicinal plants, organically grown medicinal plants. So there is no restriction as such in the land use patterns, but mostly at the moment, what is done in the country is actually wild harvesting. Now, there are restrictions for wild harvesting, especially for certain medicinal plants that has a greater demand in the other countries, like Salacia reticulata. So they are not allowed to wild harvest or transport. And even certain plants like, there is a plant somewhat similar to agarwood, where the same oil, and that is also completely prohibited. And the people who transport this also have been apprehended from police. So there are restrictions like that for the plants that has greater demand from overseas. But if you are using as the Ayurvedic practitioners or the Ayurvedic department, they can wild harvest and use it.
Yeah, sir. One more question is from Mr. Modan Harun. He said, he has asked that what challenges are faced while studying the marine pharmacology and phytochemical components.
Well, yeah, it's challenging, of course. Yeah, the thing is like, now the marine, if you take marine plants, and there are a lot of, there are a lot of symbiosis associations, so that, you will not be able to exactly tell whether it is from that particular organism or from the symbiotic organisms, and separation of them is very difficult. And also, since they are from marine system, and when you start to separate, then you face certain problems, like they are very thermally labile, and things like that can happen. And but other than that, I think you can work on that. And what you extract is also, when it comes to the concentration of those active compounds are quite low. But when you compare with the terrestrial counterparts, of course, they are very potent. So that is the advantage, although you have a very little, quantities that you can extract from. So those are the challenges. But basically, now we have been working on this for some time, and well, we have not come across. We are using the divers, of course, to get these samples collected. It is possible, but those are the limitations when it comes to marine natural products.
I would agree with Dr. Prema Kumara. Yes, the quantity to be harvested is very high, which is required because the compounds are usually present in this very small amount. Also, the structures are very complex. So, getting the right kind of structure and identification is again very difficult. And symbiotic nature is very much a big hurdle, because you will not be exactly knowing what would be the source.
Yes.
Right? Huge kind of symbiosis is observed in marine organisms. So next question is from again Malvi Vyas. She has asked that the deep details about the plant is given by Dr. Prema Kumara, that is about Salix alba. She wants to know that is there any idea of this plant phytochemical constituent salicylic acid, which is useful in the treatment of melia, that is deposition of keratin under eye, that is particularly found under the eye. And this question, basically, she is asking you whether you know that the salicylic acid is present in. She's questioning about presence of salicylic acid in Salix alba.
Yeah, of course. Now it is from Salix alba, but only thing, I have not again now studied this plant. But since it is the remarkable invention that we have made in early years, but which has it's considered as a wonder drug. And so through literature, of course, I studied this, but I have not come across anything to do with keratin. But I know that this is being used to straighten hair. The salicylic acid you use, so there may be something to do with the keratin deposition, and to curb the keratin deposition. Maybe so you have to study on this. I'm sorry, I cannot answer that straight away.
One thing I would like to add into this is this salicylic acid has been isolated from Salix alba, and these reports are of this early century. And then this salicylic acid has been converted into acetyl salicylic acid, that is aspirin, which we are regularly using as powerful analgesic, right? So the next question is what or which are the indigenous communities present in Sri Lanka that have contributed towards ethnobotanical research in Sri Lanka?
Yeah, when it comes to indigenous people, we have a community called Vedda community. So they are basically their medicinal needs are made from the wild, and they live based on the forest that they live. It's a small community. The majority is, of course, the Sinhalese, who has a long history here. And other than that, the other communities like Tamil and the Muslims who live here, they are Tamils, of course, has a long history here, but the Muslims, of course, they are traders and they came to Sri Lanka. So as early inhabitants, it is the Vedda community what we have. So there are contributions from them, of course, because they have used for certain diseases, certain medicinal plants, which are known by the the majority, and so it's been used in these studies. So other than that, actually, we don't have much history on these specific communities. But now this Rama Ravana, the story is there. So it is believed that there were communities lived in this country more than 6,000-7,000 years ago. So that is the only thing that I can trace back to the history. And in fact, I spoke to one of our archaeologists here, and he in fact he told that there are claims of living special communities during this period, and in fact has a lot of associations with Indian rishis at that time, and that those are the things that is available with us at the moment.
Thank you so much, sir. Now, next question is for Dr. Katai. Dr. Katai?
Yeah.
Yeah. Actually, this question already have been shown in this presentation. Is the propolis, that is blue, is a good drug against COVID-19, and you have shown the slide where you have shown that there are some reports of for propolis to be active against MPRO, right? You want to add into this, sir?
Nothing much actually. It's a prophylactic, someone can take to keep immunity in a good shape. So making a unproven.
One question. This propolis, actually, I have read many things about propolis. Is it used internally? I have used it in the dentistry. It is very much highly applied.
Yeah, it's used internally also. It has many applications on skin related problems. It has applications on oral, dental. Yeah, and it can be taken internally. We make tinctures, we make liquid, we make tablets, we make capsules, and Japan, the South Korea, those countries has a huge market for this. I think I saw one question.
Okay. So there is one more question. Mr. Modan Harun is asking,
I'll call you, right?
India lacks in proper functional usage of propolis. So what sort of advantages can we expect in the near future in the pharmaceutical and medicinal field with respect to propolis in India?
Yeah, that's a very good question. And before I answer that question, let me thank you for inviting me here. And it's nice to see you after so many years. And
nice for me also to have you with us.
Coming back to the question, as we all know, the Northeast region has got one state is completely organic, and they are moving towards complete organic, and they have very good plants, different plant species. So propolis collected from that region can be organic propolis sold over as a organic propolis throughout the world, and that has very good potential. And we know the Himalaya band and the Sahyadri band. So it's the longest and diverse plant flora. So propolis beekeepers from that region, if they focus on propolis, then that propolis can be a very potent source. So I think government has promoted a lot the beekeeping and the different products related with the bee. I think there are few companies who collect from and sell it to make products and sell it throughout the world from India. So definitely it is getting attention, but still I would say about 70-80% of beekeepers doesn't know the importance of these, and it has got a lot of potentials in India or the Japan or the South Korea. That kind of that side of countries can be used for marketing those products. Yeah, I think there is a huge potential. We have a number of beekeepers increasing day by day, honey production increasing, and hopefully the propolis will be more famous, and they will understand the economic benefits, and it's an addition only because they are not doing anything extra. It just taking out that resin which is on the frame, which is they are throwing as a dark sticky material now. So it is just about educating them and telling them that they can just sell that raw propolis without doing any value addition into it to the companies. I don't see there are many manufacturers of tinctures and creams and the propolis based products in India. So there are 2-3 suppliers who sell the tinctures, I guess, and the raw propolis, but not other. So as you said, and as we can see in the literature, the skin related problems can be, wound related. So propolis creams, that kind of products can be made, and there is a huge market for that. So there is huge potential which is not explored. Yeah.
Thank you so much. As such, as you said, it is a byproduct. So one need not have to go for any extra effort, right? So I think it is going to be there in near future. If someone takes up work and if the government agencies get the knowledge about this kind of product, by a product which is a byproduct actually, which is there through the production of honey by honeybee, we will be having very good market, and we'll be having many people to work on in this area too. So that is all for you, sir. Next question is for Dr. Prema Kumara. Mr. Ravindra Mali has asked you that, is there any ethnobotanical dictionary published in from Sri Lanka? Dr.
Sorry?
Prema Kumara, is there any ethnobotanical dictionary published from Sri Lanka?
Yes, we have. In fact, we have a pharmacopoeia for medicinal plants. For Ayurveda and our indigenous medicine, we have a pharmacopoeia, and also text for botanical needs. We have several actually. With the medicinal properties, we have a separate book.
And this is a general question which has been floated. Apart from composting, can dry fallen leaves be useful to extract medicines?
I didn't get you actually.
Apart from composting, can dry fallen leaves be useful to extract medicines?
Dry fallen leaves.
Yeah.
Yeah.
Asked by some participant.
Yeah, should be able to extract, bioactive molecules. Yeah, fallen leaves as such have not been explored much. It is the one that is, you usually pluck dry and then, extracts. But that's a interesting thing, that because then the maturity also matters, and, that is something very interesting to look into.
But I would like to add into this from my part that we actually see that a living tree. We always go for a living biological system as a source. So we go to the living tree, we pluck the leaves, and then we dry, and then we use it.
Yeah, that's right.
Trees are actually containing lots of tannins, and tannins are not that well worked on. And what we think is that the tree has discarded the leaves now. So maybe they won't be that useful to us. That's what is my perseverance from the idea of getting the plant collected from the living tree or a living plant, right, sir?
Yeah. Yes.
Yeah, I think that's all about the question. That is all for the question answer session. We don't have any more questions now. So I would like to thank both Dr. Prema Kumara and Dr. Katai to be with for joining with us and giving such a huge kind of information. And I would like to call Preeti online now.
Yes, ma'am. Thank you, ma'am, for conducting the question and answer session. Now, I'm sure the talks have been fruitful for all the delegates. As we can see, the chat box is flooded with numerous compliments from the participants. Thank you for your valuable feedback, participants. If any of the participants want to share his or her views live, we heartily welcome them. Kindly write a yes in the chat box. We will unmute them so that you can speak.
I would like to say a few things. This being a botanist, this was an amazing day today for me, starting with Prema Kumara sir, and then carrying on to Shankar sir, both of you, and meeting Shankar sir's little one and hearing her also. That was interesting.
Yeah.
And don't worry.
Sorry.
Yeah, hi to her.
Maybe the children they are our budding researchers and future researchers. So thank you very much for being with us today, and we really appreciate your effort to be with us. Thank you very much. And my students who are undergrad and postgrad botany students, they have really appreciated. As Preeti ma'am said, there are floods and floods of congratulations to our whole team, including you, our resource persons. Thank you once again.
Thank you very much. Thank you very much for inviting us. And I say hi to Shankar as well. And
Hi.
Thank you, Alpana ma'am.
Okay, thank you very much.
Thank you, sir.
Thank you, Professor Mamta. Thank you for inviting me. And we'll meet up slowly one day. It's a little world.
Mamta ma'am, you are muted.
Surely, sir.
Yeah, thank you very much.
Thank you.
Bye.
Thank you. Thank you.
Thank you.
Thank you, doctors.
So we are heading towards the end of the event. Please wait. So we are heading towards the end of the event. So now I would again invite Dr. Mamta Shah to give a concluding remark on the 2 days event. Over to you, ma'am, again.
Yeah, thank you, Preeti. I would like to say something about the conference, how we started. Actually, myself and Alpana madam just had some little chat about organizing one international webinar, and accordingly we planned this webinar, and she wanted it in this in a manner where a botany student or a life science student would be interested along with the pharmacy students. So accordingly we planned the entire event, and we were lucky enough to get very good speakers with us. We started with Dr. Akshay Charegaonkar, who gave us a very good detailed you can say approach how one can go for developing HPTLC method for analysis as well as qualitative analysis, quantitative as well as qualitative analysis of herbal drugs, with huge kind of examples he quoted during his talk, and he gave complete understanding to the students to students and participants about the HPTLC. And he also gave knowledge about how many labs and institutes are into this kind of research, where they are using hyphenated techniques for analysis of medicinal plants or medicinal botanicals. Then we had with us Dr. Boktiar Uddin from Bangladesh, and he actually gave he gave many examples of important medicinal plants of Bangladesh. He gave knowledge about how typical techniques of cultivation as well as harvesting of these medicinal plants, and he showed his own work. He showed the ethnic people's work along with his work, and he showed that what all are the outcome of his work with the ethnic people of Bangladesh. They have created two very good databases, and that is there on the website of Bangladesh, like medicinal plant database of Bangladesh. And it's actually very good. I have gone through that website. Huge information has been compiled on that, and it was actually a very interesting talk. Then today we have with us Dr. Prema Kumara. He actually gave stress to importance of medicinal plants. And how these medicinal plants have contributed towards drug discovery. He showed huge amount of data, ancient data as well as recent data, and he stressed significant contribution of medicinal plants towards drug discovery. He even shared his own data where they have worked on marine plants or marine organisms, and they have come up in their lab with some molecules which are active against COVID-19 too. He also have given us the examples of aromatic compounds, and one compound typically he said elimacin, which is found to be present in major amount one of the drugs. So like his talk was flooded with huge kind of information, and I am thankful to him for that. And Dr. Katai, again, he actually gave very good information about propolis. Propolis is actually very, I think many of us were not knowing about propolis, and specifically botany students. We actually at pharmacy are, and being a pharmacognosist, are exposed to many things, and I'm lucky to be there as a pharmacognosist, I would say so, because we have knowledge about like our science is like that we are exposed to many things which are from the nature, and chemistry, pharmacology, everything are been focused collectively in our subject. So propolis was discussed at length by Dr. Katai, and he gave so many, like he gave ancient how the name came up, how the propolis can be used, how honeybee is collecting this propolis, and how it can be useful to the society also. And there are many reports of propolis being used as antioxidant and wound healing, as well as it is actually very largely applied in dentistry, as far as my knowledge says. So he gave very good knowledge about propolis and all. In addition to that, on my request, he said about regulatory aspects of this medicines in UK, in particular, because UK people, they are very conservative. They don't accept these things very easily. They say what is the safety. So safety aspects are to be covered, and accordingly, these drugs can be marketed with the THR for that is certification mark is there. He talked about VHMA for a herbal mark also, and so when if you want to go to the UK market, you need to have certificate, that is THR certificate, a traditional herbal remedy certificate, and there you have to mention that since how many years this drug is used in your country. If you are an exporter from outside UK, and if you are there in UK, you have to show that it is in use in that particular country for at least 15 years. So they are very conservative. They see what all are the safety aspects. Efficacy is must to be to go in the market, but then along with that, safety is must. So he gave, I think everything was covered by him. Even he discussed about the yellow card system to report ADR, right? So his talk was also flooded and loaded with lots of information about not only propolis but also UK market, how to penetrate the UK market. So those who want to go for herbal medicine, like manufacturing of these medicines and exporting of these medicines in UK or world over, they were, I think, well informed now today after this talk of Dr. Shankar Katai. So I'm very much thankful to you too, sir. And apart from that, I would like to thank Dr. Alpna Shukla, Dr. B.K. Jain, and my principal too, like Dr. N.T. Chabaria, who always give me freedom to work. And I'm really very thankful to you all, my coordinator team, Krupa, Preeti, and even Jankna. So I hope you all have enjoyed and learned a lot from seminar. Thank you.
It was a great team effort.
Yeah, I agree with you, ma'am.
Thank you, Mamta ma'am, too. Without your guidance and constant efforts, the planning and execution of the event wouldn't have been possible. So thank you very much, ma'am. So in the end, I thank all the delegates for their keen participation and kind cooperation during the entire event and for making this event grand success. So once again, thank you all. Have a nice day. Stay safe and take good care of yourself. Bye.
Thank you.
Thank you. Thank you.
Bye. Thank you. Thank you all.
Thank you.
Thank you.
Thank you, Chabaril sir.
Thank you.
Thank you, Alpna. Thank you, Jain
Thank
sir.
you. Thank you, Mamta Ben. Let's stay back 1 minute and can we open our videos? And
Yeah,
I
sure.
request Krupa Ben to give me the highlight, but somehow she's.
Madam is only highlighted. Madam, you are
I highlighted, madam.
Yes,
I
yes.
highlighted.
I'm just, yes. Thank you, madam. I'll just take a
print. Open karo tamaro camera. Chabarya sir. Chabarya sir.
Sir જતા રહ્યા.
સરે છે ને પહેલા કરેલો.
Let me see. Let me talk to him. I will request him. Jain sir ne કોને madam? તમે?
I have told sir, I have called him also many times, but somehow he is not seeing. So he must be busy.
Okay. So let me talk to Chabarya sir. If he comes, we can have the photograph.
Madam, 1 minute, Amjol. Okay, I got that.
Thank you.
Should we wait for sir?
Just wait for a minute.
Okay.
Sir, 1 minute તમારો camera on કરો ને એક click લેવી છે જોડે. He is making his camera on. બતાવો કરજો.
Sir is coming.
Yeah. He must be rejoining, so that is the day.
આપણે હવે હાઇલાઇટ ના બદલે બધા પિન કરી દઈએ તો બધાનું ઇક્વલ આવી જશે. મારાથી નથી થતું પિન એટલે
હું કરું છું.
હા.
Okay.
સરને મેં જોઈન કરી દીધા. સર આવ્યા. છાપરિયા સર. Jain sir is still.
I request all the participants also to stay back.
Yeah, whoever wants to, and we can.
Madam, camera open karo.
BK Jain sir નથી દેખાતા અહીંયા એમને પિન કરવું છે.
એક સેકન્ડ. જંકના મેડમ ને છે ઓન છે તમારું વિડિયો?
હા, જંકના મેડમ નો ઓન છે.
સંજના મેમ છે.
જૈન સર somehow નથી, આજે કંઈક બિઝી લાગે છે. કઈ નહીં, આપણે પેલું કેવું પિન કરીએ તો આમ બધાનું ઇક્વલ આવે ને એવું કરી દઈએ.
ટાઇલ વ્યૂ આવી ગયું છે. જંકના મેમ તમારી આંગળી લઈ લો.
ઓકે.
હા,
બસ હવે બરાબર છે.
now we have this. Bhavna, Anar, Sunita ma'am, Pankti, can we request you to open your videos for a minute and then you can close, then we'll have an organizer's photo also. Shalang, Pankti, all those quickly, come on.
Hello.
Okay, I'm going ahead with this picture and then you can switch off your videos and we'll have an organizer's picture. Thank you, Dimple.
પ્રીતિ,
હા મેમ.
Yeah, thank you.
ઓફ કરેલો છે આપણે, હોસ્ટે ઓફ કરેલો છે વિડિયો એવું કહે છે. પ્રીતિ, સુનિતા મેડમ નો ફોન હતો, જરા જુઓ.
અચ્છા, સુનિતા મેડમ નું
હોસ્ટે ઓફ કરેલો છે એવું કીધું. હા તો I can do it. Ask to start video. I've asked. Yeah. સુનિતા મેડમ ને થઈ ગયો.
There she is.
થઈ ગયો, yeah.
One more picture. Okay, participants, thank you very much for joining us. Looking forward to your continued support.
185. Thank you, madam. So we shall leave now.
Okay,
ma'am.
okay.
Bye.
Yeah.
Bye.
Thank you, sir.
હું તો એક આ પણ પાડી લઉં. Madam, unmute karjo, Krupaben. I think we'll close the meeting now. Okay. Thank you all. I'm leaving. Thank you, Vanita.